Okay, so welcome. Today is the review session for the exam that will be this Thursday. On Thursday, this will be a full-length exam. It'll last the entire hour and a half. I have almost finished writing it. I'm going to wait till after tonight, see where the questions are, see if I've forgotten anything, or maybe make some modifications based upon our conversation. I suspect somewhere around 150 questions or so. And there will be quite a few of them that are very quick, and I'm going to show you some of those questions today. So you'll actually be very, very well prepared for those questions. Okay? And the content on this exam is what? Chapter 9, which was muscle. And much of that conversation dealt with skeletal muscle, dealt with the sliding filament theory. You understand how myosin interacts with actin. You know, hopefully you feel comfortable with that story. You know how the tropomyosin and the troponin and all that is interacting. There's some nice videos on that. Also, by now, you all should have done your 2 quizzes, right? So you've been through this. So I'm here to answer questions and to clarify, not to present new material. I only want to clarify and help you through this. Then chapter 9, then there's chapter 19. That was 2 presentations on the heart. You need to know your heart anatomy, right? The basics of the heart anatomy, because in order to understand the cardiac conduction system that you've been looking at in lab, and to appreciate what's going on in the EKG, you need to be reminded about the chambers and about the valves and the overall structure of the heart. And you need to be thinking about, you know, the aorta and the pulmonary trunk, those sorts of things. And then the last chapter, which I would have been presenting today, is over blood. And I'm going to try this, and I want to hear back from you if you think this works well. You know, the last lecture you get before an exam is always the one you know the least, right? It's always crammed down your throat at the last second, and you're always like, darn it, I wish I had more time. If you like this format, what I may be doing in the future is simply pre-recording the lecture right before the exam, having you do it on your own like you did, and then come together the time before the test and do a review. So you let me know if you like this review process, and if that's something you want to do in the future. I'll simply pre-record the lecture right before the test, push that earlier, have you do it, and that'll give us a good quality time for review. I typically have not been doing review sessions in my history here, so I know it's always a good thing to walk through, and so I hope that you'll benefit from this. And then the last chapter was over blood. Now you're being quiz this week in lab on EKGs and conduction system of the heart. So hopefully, along with the quiz, that's all feeling really comfortable. What you're probably the weakest on, honestly, is the most recent material, and that is the blood. So I'm going to spend a few minutes talking about what I want you to know for the blood today, and I'm going to give you a preview of about 30 points on the exam that should help you tremendously in acing the exam for that part. The blood you won't be seeing until next week in lab. Okay, so it's coming. It's really close. And I don't want to jump the gun too much, but you will be seeing blood again. So when you get to lab, you'll be in really, really good shape. Remember, too, that not only are you learning this material for the exam on Thursday, but there will be a lab exam in a couple of weeks. And that lab exam is going to go back to joints. It's going to go back to like the knee and the shoulder. It's going to go back to the nerves and the action potential stuff that hopefully is checked, got it, been there, review it, and you'll be fine. It's also going to go over the heart structures, arteries and veins, the conduction system, the EKGs, and blood. So it's a fair amount of stuff coming up in the second lab practical. So just know that as you're practicing and looking at this material, you're also, in fact, studying for the upcoming lab exam. Are there, before I get started, any burning questions? Anything you're like, okay, I came prepared with a few questions, let me ask one right now. Let's start back, grab your notes, go back to chapter 9. Flip through chapter 9. Take a few minutes. That was the muscle chapter. Most of what we discussed in that chapter was skeletal muscle, although there was a little bit of information about cardiac and smooth muscle, and I do want you to be able to compare and contrast these 3 different types of muscle. What are you looking at on the screen? Let me make this larger. This is all part of that conversation. And I'll use this as a jumping spot to discuss some things. So what are we looking at here in this image? This is a sarcomere, a portion of a sarcomere, or actually more than one sarcomere. You're seeing the sarcomere going from, what's this? This is the Z to Z. Z band to Z. You'll see Z line or Z band. It's a protein. That's a sarcomere. That's the functional unit of the muscle. And then what do we have in this image? What are, another small, what are the purple molecules? In the center, those purple represent the big molecule. That's the myosin. Okay? And in this picture, the red and green combo, that is the thin filament, right? And so what's going to happen during contraction? Actins and myosins are going to slide over each other, aren't they? And somebody talk to me about that process. I mean, go way back. I want you to step through this process, step by step by step. And you need to be able to do this in your head. So we're going to go all the way back to the axon that is sending the signal down the neuron. Okay? It's a motor neuron, right? It's a motor neuron. If this is skeletal muscle, it's going to be a motor neuron. If it were cardiac muscle or smooth muscle, then that would instead be a what? Not a motor neuron, but one of the autonomic motor neurons. Okay, remember that little distinction in the nervous system. Regardless, axon sends down an action potential. What happens? At the you're imaging, you're imagining right now that you're down at the synaptic knob. So what happens at the synaptic knob?
Calcium enters.
Yep, so here we are. Okay, I'm down at the synaptic knob. And what is going to, looks different here than it does there. What is going to happen now? I heard someone say, so here's the action potential coming down. I heard someone say, calcium's going to be brought in. What kind of channels bring that calcium in? They open because of a voltage change. So these are voltage-gated calcium channels, right? And they open up. And what happens? Those calcium ions are going to interact with the plethora of synaptic vesicles that will be down here. And those vesicles are filled with what? Acetylcholine, right? That is the neurotransmitter that works muscle. Okay. So the calcium binds to the vesicles and causes the vesicles to do what? Bind to the synaptic wall, if you will, and exocytose their products. Now, what is right here? It's a very large space there, but that synapse, this is what kind of synapse? If it's going from neuron to muscle, right, we're going to call that the neuromuscular junction. And we're going to call this area of the muscle the what? Motor end plate. It's just a fancy way of saying the area of the neuron that interacts with the muscle is the motor end plate. So acetylcholine is going to be released. It's going to be out here in the synaptic cleft. What will be here on the muscle side? An acetylcholine receptor, right? And what kind of receptor is this? It's a molecule in the membrane. It's going to receive the acetylcholine. And what happens when the acetylcholine binds to this receptor? This acetylcholine receptor actually is another gate, isn't it? What kind of gate is it? It's a sodium channel. And what kind of sodium channel? Is it opened by electrical charge? No, it opens by a molecule. We call that a ligand. So this is going to be a ligand-gated sodium channel. It opens up. The ligand is the acetylcholine. So the acetylcholine causes the gate to open. Now, what do we know about sodium? Sodium's always where in greater concentration? Outside, right? And so when the acetylcholine binds to that receptor, sodium rushes in. Now, we're in the muscle cell, right? So what's going to happen to the muscle cell? That influx of sodium is going to cause what? A depolarization, right, of the muscle cell. Where does that depolarization travel? Thank you. Where does that depolarization travel? It's going to go along the sarcolemma, right? That's the name of the outer membrane of the muscle cell. And along the muscle, there are going to be these rabbit holes. Those rabbit holes are the T-tubule. So that signal is going to go down the rabbit hole, down the T-tubule. What's on either side of the T-tubule? Cisternae. These two cisternae and the T-tubule together form the triad. So what is going to be right along this T-tubule? What kind of channel? Voltage gated. It's going to open in response to the voltage change. And when this gate opens, it's going to allow calcium to do what? It's going to trigger the release of calcium into the muscle cell, right? Now, what do we have in the muscle cell? Right? We have thousands and thousands of sarcomeres. And each of those sarcomeres is arranged along a what? Myofibril, right? Many, many sarcomeres along the myofibril. Many of these stuck inside the muscle cell. What is that calcium going to bind to? We got calcium. The calcium is going to bind to troponin. Troponin is a part of which? The thick or the thin filament? The thin filament. Can you recall what the thin filament is actually made up of? It's not just made up of actin. It's actually made up of a twisting double helix of actin, but also in addition to that, what? A long rope of tropomyosin, and to the tropomyosins there's also these little snowman molecules, the troponin. So the calcium is going to bind to the troponin on the thin filament. And by binding, what is troponin going to do? Cause, magic hocus pocus, cause shift, change, a conformational change. And what's going to be rotated now? The tropomyosin, that long cord-like molecule, is going to kind of twist. And as it twists, what's it going to expose? The binding sites for the actin. Okay? Now, what's already hanging out here right next to the actin? Myosin, right? The myosins and actins are all interrelated to each other. The myosins have those big golf club-looking heads. The myosin will now be able to bind to the actin, right? Then what happens? Let's back up. What are the steps here? Excitation, right? We've excited the membrane. The electrical signal came down. We've electrified or excited the membrane. What was the second step? Excitation coupling. So what are we doing here? We're coupling the excitation signal with the binding of the actins and myosins together. So they bind. What does the myosin do? Myosin also has within it an enzyme activity called an ATPase. And what does it do? It breaks the ATP as it does the power stroke. And now it's stuck there until what happens? What is required for the myosin and the actins to dissociate, to separate? What has to come in? An ATP. Right? So an ATP has to be available. Remember, there's a lot of mitochondria in muscle, a lot of energy necessary for this whole thing to work. And that ATP comes in and causes the actins and the myosins to dissociate. The myosin goes back and looks for another actin over and over and over. I know these videos always show you one or two of these things, but imagine just, hundreds of thousands of these actins and myosins all grabbing and pulling simultaneously, and every sarcomere within a myofibril and every myofibril within a muscle cell all contracting simultaneously. Because, remember, we've got hundreds of these myofibrils and thousands of these sarcomeres all shoved into a muscle cell. So when this signal comes down from the axon, every single muscle fiber within that muscle will be excited by the calcium, okay, and will cause this sliding to occur. That all making good sense? So what's rigor mortis? After death, no more ATP being produced. So the body goes into a state of rigor because there's no more ATP available to dissociate the actins and the myosins. Does that story from top to bottom make sense? Is there anything in that story that is not clear to you? Is that a familiar story? Am I wasting your time? Like we got this? Or are we okay with this? I'm not seeing I'm seeing confirmation, but I'm not seeing any concerns or any questions. Anything to clarify in that conversation at all? Yes, sir. Well, excitation coupling, so the idea is that you're going to couple this excitation of the electrical signal to the physical sliding of the filaments. So we have excitation, and then the coupling of this excitable signal to the actual, shortening of the muscle. It's more of it's just a term, right? It's a term, yeah. It's kind of it's kind of I don't know, it's a term that doesn't always speak to us, so I appreciate your asking that. What else was troubling? Look through this chapter, chapter 9. What else in this chapter was troubling? Let's go back to this image I have on the screen. So let's label these, shall we? What do we call these different bands? Where the myosin is, just the length of the myosin, what's that band called? A band, right? Just where the myosins are within the sarcomere. Where just the thin filaments are found. That's the I band. In the center, where the myosins are all by themselves, right? There's no actins over sliding those. In the very center of that is the H. And then the line down the middle is the M. So what is going to change in these? And then we go from Z to Z, right? From sarcomere to sarcomere. So what is going to change in length during contraction? Is I going to get shorter? The whole sarcomere is going to shorten, right? So if Z to Z shortens, then the I band would also get shorter. What is definitely not going to change? A. Right? Myosin itself does not get shorter. Actin itself does not get shorter. All we're doing is sliding them over each other. But that myosin, that A band, will not change. The I band will get shorter as the sarcomere shortens. The Z to Z will shorten as the sarcomere shortens, but the A band will stay the same. What is, this doesn't show it very well, but there's a protein that connects to the myosins from the Z band. Kind of like a big coil, like a trampoline coil. It's the biggest protein known? Titin. Yeah, titin. Okay. Any questions about the sarcomere, sliding filament, the whole story of excitation and, oh, I need to go back to that a little bit. Excitation and sliding. As long as calcium, as long as there's an action potential being sent, right? Then calcium will continue to be released into the muscle cell, and troponin will stay in its position and will allow actins and myosins to talk to each other. How do we get rid of that signal? We don't want our muscles to stay in contraction. So we need to go through relaxation. So what would relaxation involve? Voluntary muscle. You tell it to stop, right? Stop sending the signal. No more acetylcholine being released. What happens in the synaptic cleft? What's going to come and get rid of some of that acetylcholine? Acetylcholine esterase, right? ACHE. Acetylcholine esterase is going to clean it up. But also, we got to get rid of the calcium, right? Because the calcium is also floating around inside the muscle cell. So what happens? The calcium will be sucked back into the sarcoplasmic reticulum. And the sarcoplasmic reticulum is just like a silo. It's just a storage place for calcium inside the muscle cell. Once that calcium is gone, what happens? Calcium is also grabbed off from the troponin. When calcium's gone from troponin, the tropomyosin flips back and blocks the ability of the myosins and the actins to play together. Okay? We got that story? Silence is confirmation, right? Okay. Okay, let me see what else I've got here in my little images. Okay, this image, kind of switching gears a little bit, reminds me of the overall structures of the heart. This image does not include on it the conduction system. That's important. So where does the conduction system begin? If we're going to talk about the cardiac conduction system, and it's always in yellow, right? So I guess I'll go to yellow. So what it would be located up right about here? The SA node. Okay, sinoatrial node. What's that thing doing?
Pacemaker.
It's the pacemaker. These cells do not look any different from any other cardiac muscle cell. Under the microscope, you can't tell an SA nodal cell from a regular cardiomyocyte. They look the same. But there's something special or unique about those cells. That is, they don't have a resting membrane potential. They never stay at a normal resting membrane potential. Instead, they are always leaking and always moving toward depolarization. So they always are moving toward, or I should go this way for you, always going toward depolarization. How often would the SA node like to fire? Intrinsically, built in, it's going to fire how often?
60 to 100.
Close to about 100 times a minute. Okay? But the SA node and these pacemaker cells are actually slowed down a little bit in the average person because the vagus nerve, remember the vagus nerve, sends a parasympathetic signal to the pacemaker and says, cool it a little bit. So now the heart will only beat usually 70 to 80 times a minute on average. It would normally, if you severed the vagus nerve and there was no more parasympathetic signal telling the heart to chill, then the heart would normally beat about 90 to 100 times. Okay? So it's always got the vagus nerve telling it to kind of rein it in a little bit. So we have the SA node. And what does the SA node do? It fires. How often does it usually fire? On average, every 0.8 seconds. Right? We're learning that number from lab. And that means that the heart rate would be what? 0.8 seconds per cardiac cycle is a rate of 75 beats per minute. So that's around average. What does it do? That electrical signal does what? It goes out in every possible direction. It goes through the right atrium. It passes right over to the right or to the left atrium. What allows for that signal to travel so easily and so quickly? The intercalated discs, right? So that very thing that we look at under the microscope to identify cardiac muscle functionally is what allows the electrical signal to travel so easily. When all the cells of the atria act as a single unit, what's the word for that? During atrial systole, when the atria are both squeezing as a collective unit, there's a word for that. Do you know that word? Yeah. Syncytium. What does that word mean? We know we can break it down. To work together, right? Cells. So all the cells are working as a collective unit. So we would say that the atria are acting like a syncytium. They're acting as a unified group of cells. And they're all going to squeeze at the same time, aren't they? Okay, that's going to be the atrial depolarization. On the EKG, what are we going to see? P wave. Now, right about here, there's another node. That is the AV node. As the name tells us, it's at the border of the atrioventricular border. What does the AV node do? Receives that signal from the SA node and a little bit of a hesitation, and then it fires, doesn't it? So it's usually driven by the AV node. And when that goes off, what happens? This heart's not quite drawn for this, but what happens? The AV node will then send those signals down the bundle branches, down the interventricular septum, goes down toward the apex, comes back up around, and coming up around are the Purkinje fibers. The heart, when the ventricles squeeze, not only do the ventricles also act as a syncytium. Remember the left side is much more muscular, is much more forceful, but the fibers of the muscle of the heart are actually arranged more like a ringing mop. So when the ventricles contract, they don't just squeeze, they're actually squeezing and ringing. And now all that blood is being pushed up and out, either the pulmonary trunk or the aorta, right or left side. What else is going on, though, besides just squeezing and ringing, right, twisting? What else are these Purkinje fibers attached to? I'm drawing them right now. Those Purkinje fibers are also contracting the papillary muscles. And those papillary muscles, as you know, are attached to the chordae tendineae. So what is happening as the ventricles squeeze and contract? They're also yanking down on those papillary muscles. And what is then being forcibly closed? Which valves? Bicuspid or the atrioventricular valves, right? Now, what if, for some reason, those valves were faulty or did not close down completely? When the ventricles squeeze, blood would have the opportunity of going back up. And when the blood goes backwards, we call that a murmur. Okay? A murmur. Okay, we'll just use murmur. Okay, so that's the cardiac conduction system. It's not that difficult. There's not much anatomical stuff to figure out, but just definitely know SA node, AV node, Purkinje, the bundle branches, and then the Purkinje fibers, and appreciate what's happening there. And I think you're in really good shape in that from lab. I know you were in good hands last week. Okay. Any questions on cardiac conduction system? Oh, I forgot to continue this. So we said that the atria contracting was the P wave. On the EKG, what is the QRS? The largest of the deflections is caused by ventricular depolarization. Now, why is it bigger? Why is the QRS so much bigger in amplitude than the little P wave?
So the force of the contraction.
The force of the contraction. The amount of electrical activity that is being picked up in the EKG is so much greater because the ventricles are so much more muscular. So there's a lot more electrical activity going on, so the spike is much higher. And then what else is kind of hidden underneath that QRS? What do we not see distinctly on the EKG? Atrial repolarization. And then a little later on, the ventricles are going to relax. And what are we going to see on the EKG? T waves. Okay, good. We're doing okay with conduction system? I'm actually literally going through the images from the test. Okay? This is a Word document. I took the questions out, left the images in. So it's a good reminder, and I think you'll benefit from this. So we're going through some important things. Next image along the way. Oh dear, what is this? It disappeared on me. Where'd it go? There it is. What in the world does that reflect? This is what? What is that green line? Let's compare it. This would be like an action potential, right? So what is that green line? That would be the action potential of a cardiac cell. Okay? So the green, see the upward arrow? What's this number 2? Number 2 would be the depolarization, right? Rapid upward swing, sodium comes in, same old story. But what happens in a muscle, in a cardiac muscle cell that's different? What do we call this area up here? The plateau. And what was the deal with that plateau? What's different about a cardiac muscle action potential than a regular muscle or nerve action potential? It was a plateau caused by some calcium channels, right, that are acting a little differently. We never saw calcium channels in an action potential before. We only see calcium channels showing up here in the cardiac muscle when it comes to the action potential, right? We always talk about sodium and potassium. Now we're going to add calcium into the story. And those calcium channels cause or are delayed, and what that does is it creates this plateau. Now what does that plateau do? It assures that the heart muscle cells will never go to tetanus, right? There would never, because there's this delay, there would never be the opportunity for the heart to get to a place where there is a tetanus, because this delay acts as a longer refractory period and would not allow that to occur. Now, the green line is the action potential. The red line represents the actual contraction of the heart muscle. So again, there's a rapid depolarization of the cell, but there's always a delay, right? There's always a delay between the electrical activity and the actual activity of the muscle. So the red just says, okay, the heart's going to go up and come back down, and it's a much longer action potential. So another one cannot begin initially, right? Cannot begin again. So there's an absolute refractory period that gets built in here, which, like we've said, make sure that the heart would never reach tetanus. If it did, right, the heart could go into a big, we're done. What happens when the heart stops beating? We die. Yeah, right? Yeah, so we don't want that to happen, right? So this longer action potential kind of prevents the opportunity for the heart to ever get into a state of tetanus or permanent contraction. Any thoughts or concerns about this idea, this concept? This is a similar image to what's in your book, okay? Now, so I've introduced that little slide to you, and you know what that's showing, right? It's timing. The red line is the actual contraction of the muscle. Okay, so let's review this for a moment. You're being quizzed on this tonight and tomorrow night and Wednesday night, whatever, so this should be somewhat familiar. This is an EKG, pretty typical, very clean looking EKG, good for teaching. What are the waves we have in here? The first little wave, this one, P wave. Again, what's going on in the heart? Atria are depolarizing or contracting. Then there's this QRS complex. That is, again, the ventricular depolarization. And then a little bit of delay before you get to the T wave, which is the ventricular relaxation, the ventricular repolarization. You should know these normal values. In an EKG, these tracings are uniform. Each of these little squares is how much time?
0.002.
Each of the little squares is 0.04 seconds. This paper does not have it. Some of the paper last week in lab had bigger boxes. You with me? Right? There were bigger boxes, and those bigger boxes were what? 5 boxes together. And each of those bigger boxes would be, let's say, from here. I think that's right. Is that 5? From there to there, there'd be a bigger, darker line. And the big boxes would be what? 0.2. Okay. What should the normal P to R interval be from the beginning of P to the beginning of QRS? You need to know these numbers cold for the lab. I'll quiz you on it. I want you to know this for the test too. So what should that P to R interval be? 0.12 to 0.20 seconds. What does that P to R interval represent from the heart's standpoint? The beginning of P is where the heart, the atria, begins to contract, atrial systole. The beginning of the QRS is where the ventricles begin to contract. So that time of 0.12 to 0.20 seconds represents the time it takes for the electrical signal to get from where to where?
SA to AV.
From the SA node down to the AV node. If that is longer, then there's some sort of, quote, block. There's something blocking, causing a delay in the electrical transduction system of the heart. So 0.12 to 0.20. How long should the QRS complex last? 0.06 to 0.12 seconds. What is that telling you? The time for the QRS to occur is when the heart is squeezing and ringing, right? And contracting the ventricles. If that's taking too long, what might be wrong? A blockage not from the SA to the AV node, but instead a block somewhere along the bundle branches or up along the Purkinje fibers. Again, something is slowing down or completely obliterating the electrical signal to travel. Okay, it could be dead tissue, right? Could be a number of things. And then this is the average, 0.8 seconds, the time of each cardiac cycle, easiest to measure from big peak to big peak, from R to R. So you measure from R to R, and we see 0.8 seconds. How many little squares would that be? $20, right? You know, middle school math gets us, right? So 0.04, and then 20 of those, right? 20 little squares would be 0.8 seconds. So 20 little squares, right, would be 0.8 seconds. Now, a rate, if you have a cardiac cycle that lasts 0.8 seconds, what is that person's heart rate? 75. We know that, hopefully, but also to calculate it, what would you do? You won't have a calculator. Calculators are the worst thing in the world for this kind of stuff, right? You could do that, but we can't do that right here, can we?
No, you can count 3 and multiply by 2.
If you knew that that was a 6-second strip, right? Okay. But the other way, right? 60 divided by the cardiac length. So 60 divided by 0.8 gives you 75. Okay, so that's the mathematical approach, right? So you just take 60 and divide it by the time of the cardiac cycle, and that'll give you 75 beats per minute. There are a couple other methods, right, to calculate this, but it usually assumes you get a longer strip, 6-second strip. And how do you do that one? If you have a 6-second strip, and you know it's a 6-second strip, what can you do as a quick and dirty estimate of the heart rate? Multiply by the number of R waves, right, times 10 within that 6-second strip, and that'll give you an estimate. This is the other way. Another way, how many little squares were there between the two peaks here? 20. You can also do the 1500 method. 1500 divided by the 20 squares, okay? 75 beats per minute. Okay? That's higher math, right? We got too many zeros there. What's that? Do you need to count? No, you just go zip, gone. 150 divided by 2 is 75. We know that. Beats per minute. There's 60 seconds per minute, right? So if every beat is 0.8 seconds, then the units will cancel out and you'll get 75 beats per minute. The other way was the 300 blah, blah, blah method, right? 300, 150, 75. I'm not going to quiz you on that on this exam, but you will be quizzed on that this week in lab. And I think it's always important that you have two of those techniques available to you, the one that you like the best, the one that you can like the second best, and always do both of them as a backup on an exam. Check your work using the other method. Okay. Tonight, ask Mr. Mueller if you have lab tonight. Ask him to go through it before the quiz, okay, if you want to. But definitely, you want to understand this for the lab exam and for the lab quiz and for appreciating the big idea for the test. What if you had P, P, and then a QRS?
Flutter.
What does that tell you? That the signal initiated, right? The P is the SA node firing, but the AV node didn't pick it up, did it? So there's something blocking the AV node from picking it up. So that'll be a block, right? And we're not going to get into all the different types of that. But I just want you to see that abnormal EKGs are really a lot of fun to start looking at. Are we okay with EKGs? Review this, look at this. You're all going to be studying this for your exam or your quiz grade tonight, tomorrow night, or even for Thursday night's quiz or lab. So I know that you'll all look at this again before the exam. Yes?
Do you want us to identify different types of EKGs for your test?
I don't recall that. No, I don't think there's a different, like, now in the lab, you were given some arrhythmias.
Yeah.
I'll tell you right now, there's no arrhythmias on this week's quiz. But the arrhythmias will appear on the lab practical. Okay, so there are arrhythmias. There's like 6 of them, right, that you need to be familiar with, what they look like. I will tell you right now that on the lab practical, if they show you an EKG strip, it'll be the exact same EKG strip from your arrhythmia worksheet. So you'll just have to recognize those 6 different patterns. Okay. Okay. What else do I have coming up here? Aha. Okay. Any questions on heart, conduction, EKGs, heart structure, muscle, anything at all? Those are the images that I have for that. Yes, sir? Not calculate it. Okay? The question is basically this. He's asking, what is it that influences the rate of flow through a blood vessel? What were some different things that increase the rate of flow through a vessel? Is this what you're getting after, Tommy?
Like constriction and dilation?
Yeah, constriction and dilation, right? But the big one is if you're looking at a vessel, the radius, right? And the resistance is based upon the radius of the vessel. Is that what we're talking about?
Yeah, the 1 over R.
Yeah, 1 over R to the 4th power, right? So if I said to you that this is a horribly written, but if this radius is 1 and this radius is 2, how much of a difference in flow will there be? The relationship is flow is related to the inverse of the radius to the 4th power. So what would be the flow here? 1 over 1 to the 4th power. 1. What's the flow here when you double the radius? Okay, there's a 16-fold difference just by changing the radius. So just appreciate that there's a radius to the 4th power relationship between flow, and that's all dealing with the radius of the blood vessel. Flip through, flip through, flip through. Anything I'm missing on, or just major ideas or understanding on the muscle or on heart? While you're thinking about muscle, I'm going to skip over this next question and come back to it. And go all the way down past my question here. There will be something like this. Can you see that well enough? A Venn diagram, skeletal muscle, and I'll have some facts underneath it. And we're comparing and contrasting skeletal muscle with smooth muscle with cardiac muscle. This would be histological changes, this would be the use of calcium, this would be whatever, things we've been learning about. If the fact I give you is true for both cardiac muscle and skeletal muscle, you would type in or bubble in A and C. If it was something true between smooth muscle and skeletal muscle, you would bubble in A and B. If it was true of all three muscle types, A, B, C. And if it was not true of any of them, the directions will tell you to label in or bubble in D, as in dog. So for example, right, are striated. What would you bubble in? Which muscles are striated? Skeletal and cardiac, so you would bubble in A, C. If the fact said involuntary. Involuntary would be smooth and cardiac, right? So you would bubble in B and C. That's the idea. There's about 10 or 12 of those facts that are involved. How about, I don't know, is excitable? Excitable means that they would respond to a nervous signal. All of them, right? All of these would respond to an electrical signal. They're all excitable. So A, B, and C would be bubbled in. So you get the idea. Okay, so you'll have that Venn diagram. There'll be some facts about cardiac, smooth, and skeletal muscle. Much of that is review, but I want to reinforce that. Any concerns about that idea? Okay, I want you to be well prepared for that. Then finally, what I have today, the last image that I want to show you, and honestly, there are probably 25 points on the exam are from this image. Okay? And because this is what you're least familiar with, because this is the last thing you probably have looked at, you haven't taken your quiz yet necessarily for blood yet, it's not due until tomorrow. If you are looking at these images across the top, here are the questions you need to be able to answer for each of these formed elements. Name it. Now let me tell you, there's going to be a word bank for this. So you're going to have a table and you're going to have a word bank in alphabetical order. So here's what you need to know about the blood cells. Some of this, I know I'm pushing a little bit on the envelope, but I also know it's coming next week in lab, and I want you to know this. Name them. Is it a granulocyte or an agranulocyte? Those words refer to what? What kind of cells are granulocytes or agranulocytes? White blood cells, right? So you're going to ask yourself, is it a granulocyte or an agranulocyte? Remind me, please, which ones are granulocytes? Yeah, I'm hearing, is it Neb or Ben, right? NEB or BEN? So the basophils, the eosinophils, and the neutrophils. Whereas the agranulocytes are the lymphocytes and the monocytes. Lifespan. Okay, so I want you to know, how long does a red blood cell hang around? 120 days. How long does a platelet hang around? 7 to 10 days. How long does a white blood cell hang around? It's quite variable. Diameter. How big is a red blood cell? We know that one. The diameter of a red blood cell is 7.5 micrometers. How much bigger are the granulocytes? About 1.5 times bigger. So that would put them in about what size? 10 to 12-ish. Okay, 10 to 12 microns or so for the granulocytes, and the monocyte is usually 2 or 3 times bigger. So closer to 20 microns. What is the acceptable range for these numbers? Okay, I introduced that to you quickly, I know, in the presentation, but this is just white blood cells. What percentage of your white blood cells, well, first of all, let's go back to the cells. What are these things? So let's start right here. Number 7 is a red blood cell, also called an erythrocyte, right? These little tiny fragments are platelets made by what cells? Megakaryocytes, found in the bone marrow. All these cells come from the bone marrow, don't they? This big monstrous cell is a monocyte. This little guy with the eyeball looking at you is a lymphocyte. Across the top, we've got our granulocytes. If they are red granules, that is an eosinophil. If they are blue-black granules, it is a basophil. And if instead, number 1, the granules are there, but they're kind of neutral in color, it is a neutrophil. Percentage. What percentage of your white blood cells should be neutrophils? Anybody have their PowerPoints in front of them? I think it's kind of hidden. It's around page, oh, there's no pages, right? 17 point what? Say it again.
It says there's a range of 30 to 70%.
Yeah, so the neutrophils are going to be like 60 to 70%, okay? I think there's someplace else, but 60 to 70%, okay?
Oh, is it 60?
Yeah. 60 to 70% for the neutrophils. They are the most numerous. Neutrophils are the most numerous. Eosinophils. 2 to 4. 2 to 4%. And the basophils, their numbers are in the basement. They are less than 1%. The lymphocytes, 25 to 33 or somewhere in that range. Like 1/4 to 1/3 of your cells should be lymphocytes. And then monocytes are 3 to 8%. Okay, you're learning this. You're going to review this now, but you're going to know it really well for the lab next week as well. So it's going to ask you, what is the acceptable percentage of the leukocytes? Name a special characteristic of these cells. What do eosinophils do? Why do we produce higher than normal numbers of eosinophils? What is their main job? To go after what? Parasites and some allergic responses. But if you have a worm infection, your number of eosinophils will go up. So the eosinophils, these little granules, are filled with anti-parasitic things. They're going to break down worms pretty well. Basophils, those are the ones releasing histamines. So they're the ones releasing histamines when you have an allergic response. Neutrophils, they're primarily about fighting off bacteria. So those little granules are going to be releasing antibacterial factors. So the neutrophils are mostly about bacteria. The eosinophils are mostly about worms, parasites. Basophils think allergies and histamines. Lymphocytes. There's two basic types of lymphocytes. What are they? You hear about them all the time. T cells and B cells. You can't tell them apart by looking at them, but what do we know what the B cells do? B cells make your antibodies. Okay, so when we talk about antibodies, that's your B cells. And then the T cells are also a very important part of your immune system. We know from the news, right? People with HIV infection, their T cell counts go down. They can no longer fight off infection. They die, usually of an opportunistic infection. And then finally, the number of cells in 1 cubic millimeter, that is the same as 1 microliter of blood. How many of those cells would you expect to see in a microliter of blood? How about red blood cells? Millions. 4 to 6 million red blood cells per microliter. Whereas white blood cells, only 5 to 10,000. So you've got these images. And basically there will be a table. This will be, on the test, this will be a table. And you should, as a way of preparing for the blood section, definitely go back and be able to answer these questions for these different blood cells. Name them, know their numbers, know their sizes, know their abundance. And if you got that table filled out, you'll say, wow, that's an easy 25 points, Mac, thank you. It'll be matching. It'll be a word bank, and you'll put in the right value. What else about blood? What else about blood? I know that's probably where you're weakest. It's where you haven't taken the quiz yet, most of you probably. Oh, the clotting cascade? Okay. So what do platelets do? Clotting. What do platelets do? When you think of platelets, you think of blood clotting, right? And if there's any sort of damage to the endothelium, to the inside of your blood vessels, then certain signals are sent out. And those certain signals are going to cause your blood vessels to do what? What's the first thing that a blood vessel is going to do when it's damaged? Constrict, right? Makes sense. If you have damaged the blood vessel, you want to minimize the blood loss, and so the vessel will automatically constrict and therefore reduce the flow of blood through that damaged vessel. The damaged vessel will also release some products that will, quote, activate the platelets. And the platelets will degranulize. They will release their products. And those products are going to start the cascade of clotting. And there will be first more of a soft clot where the platelets will aggregate. And then what other protein is part of the clotting process? What protein is floating in your blood all the time, but is soluble, does not come out of solution? And then becomes insoluble when it's cleaved. Do you remember this story? Yeah, so let me go to a clean screen. So what is this protein that is floating in your blood all the time? Fibrinogen. It accounts for about 4% of your blood proteins. Well, I'm talking about blood proteins. Where are all your blood proteins made? Where is the albumin made? Where is the fibrinogen made? In your liver. Yeah, so the liver has an important function in your blood. Normally we think of the liver making bile for digestive products, but the liver is also filtering your blood, and the liver is also making fibrinogen, making albumin, making the proteins of your blood. A person with liver damage, the liver wasn't healthy, they would have problems making these proteins, and their blood would not be forming naturally. So what happens when the blood clot begins? Fibrinogen is cleaved, the protein is cleaved into fibrin. Fibrin is a smaller protein, but what fibrin does is it sticks to itself and it makes basically a clot. And all these molecules stick to each other when you make fibrin. And that is going to catch red blood cells, platelets, right? white blood cells, all that's going to get caught up in a web, and that's going to stay there for a while. That's going to, the blood vessel has already constricted its flow, and it now forms a clot. We don't want that clot there forever, though, do we? So after a while, what's going to come along to clear out the clot? There's two molecules I'm going to mention, plasmin and thrombin. Tell me about those. Turn to that page. Plasmin and thrombin. Somebody tell me the story. After the clot is made, right, we want to restore the flow after a while. What does it tell us, Roxana? I know.
It will complete the process.
Thrombin does what? Converts fibrinogen to fibrin. That's the molecule that actually did this, right? So thrombin is what does this. And then what's plasmin? Plasminogen, right? It's a protein that's floating around that's not activated. Plasminogen is activated to plasmin, and what does plasmin do? Starts to break down the clot. Okay. So thrombin is a protein. It ends in IN. It also happens to be an enzyme. It doesn't end in ACE, right? But think of it as thrombinase if you want to, just to remember it. But what is that molecule to? Thrombin is what cuts fibrinogen into fibrin and starts the clot. And then to get rid of the clot, we're going to take plasmin, and plasmin is going to start eroding the clot. We don't want it there forever, right? So this is all about damage control. So we're going to try to stop the bleed naturally. Now there's also a bunch of factors involved, and I didn't get into all the details of this. Factor 8, factor 7, factor 10, factor 3. These factors are each necessary for the clotting process to occur. They were numbered by Roman numerals based upon their order of discovery, not in the order of their use in the pathway. So I'm not going to ask you to know the order of these numbers. They kind of jump around randomly. But basically what happens, there's two different pathways of clotting, and they merge together in a common pathway, the intrinsic and the extrinsic. And basically one is happening from within and one is happening from the outside. Intrinsic, right, it's happening from within the vessel. Extrinsic happening sort of from outside signals. But they work together to form the common pathway and together create a really good, strong clot. But what if you're hemophilic? If you have hemophilia, you're missing one of the clotting factors. So if for some genetic reason you are missing factor 8, which is the most common form of hemophilia, then you would not be able to clot blood. You wouldn't be able to do the cascade of clotting, and people bleed, right? So people with hemophilia, they have a minor cut or a minor bruise. I mean, they just bang their hand against the table, they're going to have internal bleeding, right? And it doesn't heal. It doesn't clot. And so just a little bit of damage can be fatal for those individuals. So what do those individuals have to get? Transfusions. They have to get plasma. So we go down to the plasma center, right? We give plasma, and we're giving our proteins of our blood away, and those clotting factors are part of that.
What is
Vitamin
vitamin K?
K is also a part of the cascade. We check vitamin K in newborns, right, to make sure they have the proper cascade for clotting. And vitamin K is also made in your gut by bacteria. So our normal flora, our normal gut bacteria help make the vitamin K, but it is a part of the normal cascade.
Before my daughter had her liver transplant, she needed vitamin K injections on a
Again,
regular basis. Is
yep.
it something that, I know it's a fat-soluble vitamin, but does it have anything to do with these particular,
It's definitely involved in clotting.
but not necessarily in like the production of these kind of,
Not offhand. Not offhand that I can remember. Yeah. Sorry.
I was just curious.
There's a lot to vitamin K, and I'm forgetting on the whole story, so clearly you don't need to know it very much, right? Just know vitamin K.
What was one, I
Yeah.
don't remember seeing it in here, but I remember answering questions
Yeah.
on
No, it is a fat-soluble vitamin. A, D, E, and K are fat-soluble, but other than that, it's just a part of the blood clotting cascade. Okay, other questions for me? I'm trying to think through the test. I should have brought a copy of my draft. About 20 true-false questions. Okay. Lots of multiple choice.
On the blood disorders,
Mhm.
how much are we gonna have to know about, I don't know how to pronounce it,
Thalassemia?
it doesn't
Okay.
give a whole lot of information on that.
So what are let's talk about hemoglobin, okay? So what does hemoglobin do? Carries oxygen, pretty important molecule. How many oxygens can bind to a hemoglobin? 4. Sickle cell anemia, right, is a disorder. We'll talk about a couple of disorders. Sickle cell anemia is a disorder where what happens? What's messed up? The hemoglobin, right? So the hemoglobin's messed up. And as a result, what happens? Rather than the little hemoglobin molecules just behaving themselves in the red blood cell, they begin to polymerize. They make a long chain. And that long chain does what? Changes the shape of the red blood cell. We already know a red blood cell can change shape. Remember last semester we talked about hypo and hyperosmotic. So we know that tonicity, we know that blood cells are able to swell and to shrink, and that long chain of hemoglobin molecules actually changes the cells to become sickled in shape. Okay. And not only do they change shape, but that is now too big to get through most of your capillaries, and these hemoglobin molecules are not as well adapted to carrying oxygen. So that's a little bit about sickle cell. Now, thalassemia is the hemoglobin molecules, 2 alphas and 2 betas. And there's different kinds of thalassemia. There's alpha thalassemia, beta thalassemia. These are all disorders of hemoglobin. Okay, so just know that their difference is in hemoglobin, and if you don't have enough of the alpha or enough of the beta subunits, then you would not have enough hemoglobin, and you would have a difficulty in carrying oxygen to your tissues. Thalassemia is more common in Mediterranean populations, and sickle cell is more common in African populations, but not exclusive. What other disorders were in there? Okay. Thalassemia was mentioned, sickle cell was mentioned. Pernicious anemia. What does pernicious mean? Anybody know? Deadly. So pernicious anemia is a low enough level of anemia. What's anemia? What do we call a low number of red blood cells? Or anemia, right? What do we call a high number of red blood cells? Polycythemia. Too many cytos, right? Condition of the blood. So too many red blood cells. Polycythemia. So pernicious anemia is just a situation where the anemia is so bad it could cause death. Now, usually pernicious anemia is from a long-term deficiency in B12. B12 is one of the B vitamins. And we don't see this very much anymore. But B12 is actually made available in your body from a factor in your stomach called the intrinsic factor. So where we see some anemia now, well, there's all sorts of anemia, right? Just from menstrual cycle or low iron in your diet. There's lots of reasons why someone can be anemic, but also a B12 deficiency. So B12, you need the intrinsic factor. Intrinsic factor is made only in the stomach. People who've had a gastric bypass procedure no longer have enough intrinsic factor made so that in their diet they don't release B12. B12 is necessary for your hemoglobin to form. So it's one of those necessary cofactors to get hemoglobin to form. So people with gastric bypass usually need to have injections of B12 on a regular basis because otherwise their numbers drop. And it become a big problem with anemia and not having enough. They just feel kind of lousy. They get a B12 shot, they feel better. So there's a few of those different disorders like that.
Is it true that B12 is not
It's not easily absorbed through the gut, so that's why they usually get shots. You can do the lozenges and stuff, but the absorption is low. So it's better to get the shots if you need B12. Anything else from blood?
You want us to know about the genetics?
Not so much the genetics. It'd be not really a genetics class. Which question are you thinking about specifically?
Well, just about
Blood typing?
Like the RH negative and the...
Let's talk about blood typing. Because blood typing, I mentioned it briefly, this should be in your back pocket. But is it?
We had AMP for...
Okay? There's a red blood cell. That red blood cell is of A positive type. What is it that makes a blood cell an A red blood cell? It's because you have inherited from your parents genes that make the A antigen. The A antigen is a sugar group on the surface of your red blood cells. If you have the A antigen, that means that on the surface of your red blood cell you have the A. Okay? The positive means that you have also inherited from your parents what's called the RH factor, or the D antigen. RH comes from its discovery in rhesus monkeys. We share the same marker with rhesus monkeys, and so it's called the RH factor. It still sticks. So if you have A positive blood, not only did you inherit the A antigen, you also inherited from your parents the RH factor. What did you not inherit? B. Because in the ABL blood system, right, the most common blood system, we're dealing with 3 antigens on the surface of the red blood cells. The A, the B, and or the RH. Those 3 things on the surface dictate your blood type. So if you don't have B on the surface, that means your body would make antibodies against B. Okay, so your body would reject B. There would be antibodies in your body made randomly that would fight off the B antigen. The problem with that is what happens. If you receive the wrong blood type, what happens? If you are type A positive and somebody mistakenly gives you a B infusion, it's not good. Why? The person's making antibodies against B. And the antibodies will bind to and clump with the foreign B blood cells. That clumping is what the immune system does. That's what antibodies do. When something foreign comes into the body, antibodies clump on it, and that activates the immune system, and the body carries it away and gets rid of it. The problem is, this clumping is happening inside your blood vessels. And that clumping can block your blood flow. If that blood flow is blocking to your brain or your liver or your kidney or some other vital organ, this could be death. Or it could be a lot less severe, depending upon how strong your immune response is and how much of the wrong blood was given to you. So what sorts of blood can you not receive if you are A positive? I'm going to put an inward arrow with an X through it. What can you not receive? Anything with B in its name. So you would not be able to receive a B positive or negative blood, and you would not be able to receive B positive or negative blood. They all have B in their name. You can't receive them. If you received them, they would clot, and you could potentially kill the patient. What can you receive? You can receive the A's and the O's. O is unique. O is called the universal donor, right? Everyone can receive O negative. And then when you give your blood away, to whom can you give your blood? What are you giving away? A and RH. So you have to give it to individuals whose immune system is okay with receiving both A and RH. What blood types have both A and RH? A positive and AB positive. So this person could give to either of those 2 groups. I hope that's oh yeah, I know that. Okay. I hope so. There's a couple questions. There's a question or two on blood typing. Is this going to be a fun test or what? Okay, yeah. Okay.
Sensitization. I guess I didn't, maybe I read it wrong or I'm not understanding, but are those antibodies already there or do you have to have an introduction in your blood?
Your body, this is a magical story that I have not covered, but your body, through a random process, will make antibodies against every possible stinking molecule on the earth in a random process. And it will, once it's made those antibodies, if the antibody is against yourself, it'll be destroyed. So the only antibodies your normal individual will have are against things that they do not themselves have. Some things, though, do require sensitization, which is why we get a vaccine. Right? We get a vaccine to make our body more sensitive to an antigen in the future. And in the case of RH, typically there has to be sensitization. So what is the disease that I mentioned with RH factor? HDN, hemolytic disease of the newborn. And what is that disease? What's the story with that disease? Mama's pregnant. Mama is what blood type? Negative. A negative, B negative, O negative, AB negative, any of the negatives. She does not have the RH factor. Daddy, however, was RH positive, and baby has inherited that RH factor from daddy. So the baby's RH positive. No problem. Right? Mama's negative, baby's positive for RH, no problem. Baby's born, no problem. The problem is birth's a little bit of a bloody mess. And when the placenta ruptures, there's always some of the fetal blood getting into the maternal blood. So now what has the mother been given? Basically a vaccination of RH positive blood. Okay? She's fine. Baby's fine. No problem. Next baby, if it is also RH positive, these antibodies can cross the placenta. And the mother's antibodies against the RH, right? We just kind of vaccinated her during the first pregnancy. Then those antibodies can cross over the placenta and actually attack and destroy the baby's red blood cells if the baby's RH positive. So this was historically a really big cause for great-grandma having 15 pregnancies and only having 5 births, right? A lot of babies were lost to RH factor issues. They were just miscarried early in pregnancy, right? This was the common thing. Now what do we do? Any gram-negative mother, any RH negative mother, right, is going to receive during pregnancy and then right before and after birth a shot of Rogam. It's just basically I think of it as like a sponge. It's going to suck up all of the baby's blood cells and prevent her from becoming sensitized to RH factor so that the next baby who's RH positive won't have the mother's antibodies fighting it and everything is good. Doesn't matter who daddy is, right? If mama's negative, we're going to get that shot. And that's part of our normal prenatal care. Life is good in this part of the world. Other parts of the world, though, where they don't have that technology or they don't see it often, HDN is still a big problem. Does that clarify? Okay, about HDN. Okay, so anything else? We got a few more minutes. Anything else? Flip through your notes. Anything else at all?
We have to know about the different blood pressures, the osmotic pressure, and the
In the capillaries, right? What's happening in the capillaries? That's a good conversation to have right now. Oh, you guys know I can't draw. Okay. So this would be an arteriole bringing blood into the capillary. And this represents the capillary bed. What happens here? This is under pretty good pressure, right, going in. So what happens to water and small molecules? They're going to be pushed out, right? They're going to go out of the capillary through the capillary, and they're going to go into the interstitial space around the capillary bed. But what happens over here on this side of the capillary? much of it is going to be reabsorbed. Now, what is it? We learned this in Lab 2 in 105. Solutes suck. Okay, so as water is being pushed out of the capillaries, what is not being pushed out? Proteins. The most abundant protein are the albumins. So the proteins are not being shoved out. So what's happening to the concentration of proteins here in the capillary bed? As blood goes through, the concentration of proteins is increasing. And that protein is going to start sucking the water back in. So about 85% of the fluid that gets sent out of the capillary bed is going to be reabsorbed at the distal end. The other 15% that does not get sucked back in will be going where? To the lymph, right? The lymphatic vessels are sitting right there. They're going to carry that fluid. It's going to become part of the lymphatic fluid. It'll all come back, right? All these things come and connect together. What are the names of these pressures? What's pushing the blood into the capillary and out? Hydrostatic pressure. Hydrostatic pressure, the pressure of the water, right? It's the blood pressure. Hydrostatic pressure is the pressure of the blood coming into the capillary, and that's what forces it out into the interstitium. But there's also an oncotic pressure. The oncotic pressure is the pressure of the proteins. So cross off the word oncotic and just put protein. I hate that word, right? It doesn't mean anything to us. But the oncotic pressure is the pressure that's pulling the fluid back into the blood on the distal end of the capillary. So you've got the hydrostatic pressure pushing out, you've got the oncotic pressure pushing in. There's a balance. Now what if that balance gets out of whack? What happens in your interstitium if too much fluid is leaving your capillaries and going into your tissues? Edema. Right? And so if you have edema, you have too much fluid out in the interstitium. Have you seen those kids from Africa who are starving and they have the big bellies? Why do they have the big bellies? They're not starving. They've got big bellies. You got skinny little arms. They have big bellies. What's all that fluid? It's edema. Why is that water accumulating in their gut and not being reabsorbed into their bloodstream like it should be? They don't have enough protein in their diet, so they can't make enough albumin. If they can't make enough albumin, the proteins can't suck the water back into the bloodstream. Okay? So that hopefully that picture will help make sense of that pressure and why the proteins are so important in the blood. They're there for all sorts of reasons, but they're also there to help regulate that flow of fluid in and out of the capillaries. Okay. If you have any other concerns, please email me. What's still left to do? There's still a quiz for blood. And homeworks ideally are done, but you can always, go back and look at those. If you have any other concerns, though, please come talk to me.